now.
Okay.
So,
And Matt's mics as well. Does he have a separate mic or?
Right there. All right, great. Thank you. I can kind of pull that out of the way a little bit more for you as well. All right, great.
Thanks so much, of course. All right, good afternoon. It's a great pleasure to introduce today's speaker, Dr. Matt Spitzer. So Matt is an associate professor in the Department of Otolaryngology, head and neck surgery, as well as the Department of Microbiology and Immunology at UCSF. Matt received his PhD at Stanford, working with Gary Nolan and Edgar Engelman in 2016. Since then, he's rapidly progressed from a Parker faculty fellow to assistant professor, and he is now the Werby associate professor. Matt has been prolific and has really done pioneering work in the field in multiple areas of immunology, but particularly tumor immunity and immunotherapy. And I invited Matt because I think he's a great example of how you can merge experimental biology techniques with computational biology approaches, as well as cutting edge single cell approaches to really gain new insights into fundamental biological problems. Matt's clearly a rising star in the field. He's funded by multiple agencies, including but not limited to the NIH, the DoD, the Cancer Research Institute, American Cancer Society. And he's also been recognized by multiple accolades, including the Lloyd J. Old Star Award from the Cancer Research Institute, the American Society Research Scholar Award, and the DoD Era of Hope Scholar Award. As a token of appreciation for Matt joining us today, we went through lots of extra effort yesterday to get here with the bad weather. We would like to give Matt this Cancer Warrior Award, which is in recognition of his outstanding contributions to cancer research and his contributions towards mentoring the next field of scientists.
So much. Thank you.
All right, thank you, Matt. Really appreciate it.
Well, thank you so much for the incredibly kind introduction and invitation to join you all today. It's a real pleasure to be here to visit with lots of colleagues and see your beautiful campus. So thanks for hosting me. I'll tell you today about a couple stories from the lab. Oh, first, because this is a CME event, I'm supposed to show you this slide. If I can figure out how to advance the slides, my mouse seems to have disappeared. Oh, okay, great, easy. So these are some disclosures. The relevant one is with Genentech. I will show some data from a clinical trial that they sponsored and we analyzed tissues from. So I'll tell you a couple stories today about ongoing work in the lab where we really try to understand how the immune system makes decisions. And we think about this primarily in the context of tumor immunology, where the immune system can decide to either be tolerant to a tumor or to reject a tumor. We think a lot about these different tiers of organization that lead to decision-making within the system. So at the organismal level, we know that the immune system regulates virtually every physiological process in the body. And when it senses deviations from homeostasis, tries to return the system to that state. At the tissue level, we know that immune cells constitutively infiltrate every tissue across the body. And while these cells are distributed across really large anatomic distances from one another, they still somehow need to communicate and coordinate their responses to form coherent responses. Engaged in what type of immune response. to elicit in response to different types of perturbations or stimuli. And so that's been a major focus of ours, thinking about individual cells and how they're behaving and how collective decision-making takes place. And the first story I'll tell you about is something that we've recently published, and it's focused on this question, which is, where do T cells get activated after immunotherapy? So we've spent a lot of time trying to dissect how immunotherapies work mechanistically, which immune cells are involved and how are they involved and at what sites are they involved. And a principal reason for this is that we know, for instance, that T cells initially get activated in draining lymph nodes. This is common to infections and autoimmunity as well as cancer. And when T cells see their foreign antigen in the lymph node in the context of a stimulatory context where they also get co-stimulation and cytokine signals, they will clonally expand, proliferate, and many of these cells will go on to egress through the afferent lymphatics and into the circulating blood, where they can go to any site in the body, guided through inflammatory and chemotactic cues, and then extravasate into various tissues to carry out their effector function. And in the tumor, of course, the primary site here would be the tumor microenvironment, where the cancer cells are present and the tumor antigen is present. So there's been a lot of interest by us and others to understand, when we give immunotherapy, are we engaging populations of T cells that are residing in the lymph node, or are we engaging populations of T cells that are residing in the tumor microenvironment? And the answer to that question might steer how you'd think about targeting those cells, what molecular pathways might be keeping those cells in check, and what other cells they may be interacting with that regulates whether immunotherapy is effective or ineffective. And the reason we really got interested in this question was from a study that we did a number of years ago in a spontaneous mouse model of breast cancer, where we treated mice either with an effective immunotherapy or an ineffective immunotherapy. And we asked a relatively simple question, which is, what makes these contexts different? We did this by applying some single cell tools that we had developed at the time. We analyzed all of the tissues we thought might plausibly be relevant in regulating the anti-tumor immune response. And from all of those cells, analyzed individual cells using a technique called CyTOF, or mass cytometry, which we had spent a number of years working on developing. And so the question was, really just that. What are the differences that we see in the way the response is coordinated if the immune system is rejecting the tumor or if it's not? And I'll just show you one slide of data from this study, which is looking at where immune cells clonally expand and proliferate after effective immunotherapy. So what we're looking at here are two graphical networks. Each node in the network represents a cluster of cells that was grouped together based on their similarities in protein expression. So you can think of that as a cell subset. And it's colored based on whether it changes its proliferation rate after the effective immunotherapy is given. So if a cluster is colored in gray, it doesn't change its proliferation rate after immunotherapy. And if a cluster is colored in red, it statistically significantly increases its rate of proliferation as measured by Ki67 expression. And so hopefully what you can see is that in the tumor microenvironment, there's really very little change in proliferation, regardless of whether the animals were treated with the effective immunotherapy or not. But in the tumor draining lymph node, we saw that there was really profound proliferation of a number of different cell types. And this actually lasted for quite a while. The data that I'm showing here are from 8 days after the immunotherapy was given. So we still see this prolonged proliferative response in the lymph node. And we saw evidence of these cells also in circulation. And so that led us to this question of, where here we were able to block the egress of immune cells from the lymph node by giving a small molecule called FTY720 that binds to the sphingosine 1-phosphate receptor, which is the pathway that immune cells use to egress from secondary lymphoid tissue. So by blocking this pathway, we could prevent cells from leaving the lymph node and ask the question, if you block that trafficking, does the immunotherapy still work? And the answer here was relatively simple. You can see in the blue curve, animals treated with immunotherapy alone, they control their tumors. In the red curve, if you also block egress with FTY720, the tumors continue to grow out. This was with a relatively complicated immunotherapy that I won't go into today for the sake of time, but we also to know if this was true for other types of therapies like checkpoint inhibitors. And particularly, we turned to anti-PD-1 because the thought at the time really was that PD-1 is most highly expressed by exhausted T cells. Those reside in the tumor microenvironment. And so anti-PD-1 is probably having its major mechanism of action in the tumor. And we were surprised to find the exact same result, that if we blocked egress from the lymph node, anti-PD-1 had no effect in a different mouse model that is in fact responsive to anti-PD-1. So this really suggested to us that this lymph node response was pretty critical for mediating anti-tumor immunity. And right around the same time, several other groups actually published very similar results from similar experiments. So that seemed to be reproducible, which is always great. So just to summarize kind of the background here, what we had found is that T cell proliferation primarily takes place outside of the tumor microenvironment after effective immunotherapy, and it's sustained in the lymph nodes. And we found that those responses outside of the tumor are really critical for the immunotherapy to be effective. Now, at the same time that we were doing those studies, there was really important pioneering work done by a number of groups, including John Ware's lab at UPenn, from whom I adapted this slide, understanding the state in which CD8 T cells are present within the tumor, and understanding that there were a lot of similarities between tumor-infiltrating CD8 T cells and those that take place or expand in the setting of chronic viral infections. And this is a program called T cell exhaustion, by which T cells downregulate their effector function over time when they're chronically exposed to antigen. And what had been described in the literature in the context of chronic viral infection was that there's a hierarchy of these exhausted T cells that maintains this lineage, that starts with a stem or progenitor-like population that, for the sake of the talk today, I'll call TPEX for progenitor exhausted cells. And these co-express PD-1 together with a transcription factor called TCF1 that gives these cells some stem or proliferative capacity. They can self-renew. When these cells get activated, they give rise to a transient population of cells that I'll call intermediate exhausted cells for the sake of the talk today. These cells have now downregulated TCF1. They're often proliferative, express Ki67, but they haven't yet undergone the process of terminal exhaustion, although they are destined to do so in the absence of signals that would deviate them from this trajectory. And so they ultimately do become these terminally exhausted cells that upregulate other inhibitory receptors like CD39 or LAG3 or TIM3. And these cells actually undergo permanent epigenetic modifications that are not reversed by checkpoint inhibitors. And so in the context of chronic LCMV infection in the mouse, the major population of cells that actually proliferates when you give anti-PD-1 are these stem or progenitor-like cells, and the terminally exhausted cells really don't and don't reverse those epigenetic modifications that they've undergone. And so we thought, maybe something similar is happening in cancer, where there could be a stem or progenitor-like population. Perhaps it's enriched in the lymph node, and if that were true, that might help explain a lot of the conflicting data in the literature. At the same time, there was more evidence from human studies that immune cells from outside of the tumor could be important. This was an important study from Howard Chang's group at Stanford, where they looked at pre-treatment and post-treatment biopsies from patients with basal cell carcinoma receiving immunotherapy. And they used single-cell RNA sequencing paired with TCR sequencing to ask about the clonality of the activated T cells after treatment. And the conclusion they came to was that the majority of activated T cell clones in the tumor after treatment could not be found in the tumor before treatment. And so they termed this clonal replacement, which is, again, pretty consistent with this model where a lymph node response might be driving the ultimate T cell response to therapy. And there were several studies looking at immune responses in peripheral blood, which of course is the tissue matrix that connects the lymph node back to the tumor site, identifying that expansion of effector memory-like or intermediate exhausted-like cells in peripheral blood was highly associated with response to checkpoint inhibitor treatment in a variety of different human patient cancer cohorts. And so with that as background, we underwent a study in head and neck cancer to understand the relationship between the lymph node and the tumor microenvironment in patients with cancer. And we chose head and neck cancer for the specific and simple reason that when these patients are treated, surgery is utilized as a primary treatment modality. And during these surgeries, oftentimes, most frequently, patients are treated with nodes and tumor samples from patients undergoing standard of care surgery. These patients had not been treated with immunotherapy. And we asked, how do the CD8 T cells in the lymph node compare to the CD8 T cells in the tumor microenvironment? And we did this by CyTOF. So we used an antibody panel that was very focused on T cell biology, stained cells from these different tissues, and then analyzed the different CD8 T cell subsets that were present. And so from the CD8 T cells, we identified 20 different clusters that had different molecular features within the CD8 T cell lineage. You can see on the left here a heat map that delineates and annotates what each of these clusters is relative to our conventional understanding of T cell differentiation. And we asked which of these subsets is differentially abundant, either in the lymph node or in the tumor. In the tumor, we found that the terminally exhausted subsets and tissue resident subsets were more abundant, and that's pretty much what we would have expected to see. And in contrast, in the lymph node, we found that there were a few clusters that were enriched, but 2 of the 4 were progenitor exhausted CD8 T cells. So they co-expressed both PD-1 and this transcription factor TCF1, very similar to the population that had been described in chronic LCMV in the mouse, as well as in mouse tumor models. And shown here at the right is just the detailed analysis of the frequency of those 2 clusters across individual patients, where you can see that the TPECs are enriched in the lymph node from these paired samples. So we then wanted to know whether there was any relationship between these TPECs in the lymph nodes and the T cell response in the tumor microenvironment. And to do this, we performed a study similar to the one I described from Howard Chang's group, where here I'm single cell RNA sequencing paired with TCR sequencing. So we could ask whether the cells in the lymph node were clonally related to the more terminally differentiated cells that we saw in the tumor microenvironment. So we again clustered the cells and annotated them and looked at shared clones where we could find the same T cell receptor sequence present in the lymph node and the tumor, and then analyze the differentiation state of those cells. And indeed, that was what we found. The cells that were present in the lymph node had higher expression of a progenitor exhausted gene signature that had been described in the literature. And conversely, their counterparts that were clonally related, they had the same TCR sequence, but they were instead found in the tumor microenvironment. Those cells had a more terminally exhausted gene signature. So we then wanted to ask what happens when patients are treated. Are these cells in the lymph node actually responding to treatment? And to do this, we opened a small neoadjuvant clinical trial at UCSF where patients with head and neck cancer who would have been treated with surgery as standard of care opted in to get 1 or 2 cycles of a checkpoint inhibitor, in this case anti-PD-L1, atezolizumab, prior to their surgery. And the idea here is that perhaps by stimulating a T cell response prior to the surgery, you might prevent tumor recurrence, or you might allow a strong T cell response to, in some cases, even clear the tumor before the surgery takes place. And so 9 patients were enrolled in the study from whom we received tumor tissue and lymph nodes at the time of surgery. And we contrasted those lymph nodes with 22 patients that were treated at UCSF over the same interval of time and were matched for demographic variables, but did not receive immunotherapy. So we call these the standard of care cohort. And from these individuals, we analyzed both uninvolved, meaning there's no metastasis, it's a healthy lymph node, lymph nodes, as well as metastatic lymph nodes where there was tumor involvement. And the way that we analyzed these, because we were also interested in the spatial relationship of cells, was to take tissue cores from those blocks, construct a tissue microarray, and then we analyzed them using a spatial analysis approach called multiplexed ion beam imaging, or MIBI. And the way MIBI works is very similar to CyTOF in principle. So we use antibodies that are conjugated to heavy metal reporter tags. And we quantify their abundance in each pixel of tissue by using mass spectrometry. So the way this works in general is start with a tissue biopsy. In this case, it was our tissue microarray that contained lots of little cores of tissue from all of the patients that were treated as part of the study. And we stain those tissues then with a panel of antibodies. We can measure about 50 antibodies at a time using this approach. Each of those antibodies is tagged to a different metal isotope that has a different molecular weight. And then we interrogate the tissue pixel by pixel using a primary ion gun that liberates those metal ions that are bound to the antibodies just within that pixel of tissue. And we can suck up those ions and send them to a mass spectrometer to quantify how much of each metal was present in that pixel of tissue. And from that, it becomes a measurement of how much of that protein was expressed in that pixel of tissue. And ultimately, we can segment the cells within the image to define the boundaries of the cell and say how much of that protein was expressed by each individual cell in the tissue. And so by doing this, we construct an n-dimensional image. Here it's about 40 to 50 dimensions, depending on how many antibodies we measure, at subcellular resolution, and really see what each individual cell is doing in the tissue. And so we started by looking at the CD8 T cells again, and we asked, what subsets of cells do we see changing in their abundance or in their behavior between the patients that had been treated with the checkpoint inhibitors compared to those that were just treated with surgery alone? And so on the left here is another heat map showing you the protein expression in each cluster of CD8 T cells that we identified in the data. In general, the phenotypes here are very similar to what we had identified by CyTOF. And when we looked at which cell subsets were different between the treated and untreated patients, we found that the subset that decreased the most in frequency was actually these progenitor stem-like CD8 T cells that co-express PD-1 and TCF1. They were at lower abundance in the patients that had been treated with immunotherapy. And in contrast, the population that had increased the most in frequency were PD-1 positive cells that had no longer expressed TCF1, but also had not yet upregulated markers of terminal exhaustion like TIM3 or CD39. So this matches to that intermediate exhausted phenotype. And those were elevated in frequency in patients that had been treated with immunotherapy. So if we then quantify a ratio between these two PD-1 positive subsets to say what's the overall differentiation state of the cells, we see that there's a significant difference. And in general, we see more of the differentiated intermediate cells and fewer of the progenitor cells in the lymph nodes of patients that had been treated with atezo. So that would be consistent with a model in which atezo is activating the progenitor cells by blocking the inhibitory signals they receive through PD-1 and allowing them to start to differentiate into the intermediate exhausted cells. We also saw that there was an increase in the proliferation of those intermediate exhausted cells. And because this was spatial data, we could also ask where these cells were physically located in the tissue. If one was giving rise to the other, you might imagine that they would be in spatial proximity to each other because they're differentiating. And that was exactly what we saw. So if we looked at each individual progenitor cell in the lymph node and asked how many intermediate cells do we see surrounding it, we saw a significant increase in intermediate cells surrounding each TPEX in the patients that had been treated with atezo. So we then wanted to know what might be the cells that are providing the activating signals. Anti-PD-L1 is taking away an inhibitory signal, but it's not providing any new activating signals to the T cells. And the obvious candidate here would be dendritic cells, professional antigen presenting cells that can cross-present antigen to CD8 T cells. And that was consistent with what we saw in the data. So if we looked at where the TPEX were localized, we saw that they had more dendritic cell neighbors after treatment than were present in the standard of care. So there are now these niches that we had identified that contain dendritic cells, progenitor exhausted cells, and intermediate exhausted cells that tend to co-localize together after treatment. And consistent with the fact that those might be providing activating signals, if we looked at the TPEX or the intermediate cells just that were neighboring those dendritic cells, we could see that those cells now had upregulated expression of PD-1 in the lymph node, which is induced by T cell receptor signaling. So that would be consistent with them having recently received activating signals. And at the right here is a representative image of what this looks like in the lymph node. You can see that action here is predominantly taking place in the paracortical T cell zone. That dark circle in the middle of the image is a B cell follicle. And we don't see that that's where the progenitor cells are. They're mostly in the T cell zone of the lymph node. But we can find them there in close proximity to CD11C expressing dendritic cells. And we can see them proliferating by the fact that they express Ki67 in the nucleus. So that was telling us that, the lymph node biology we were seeing here made some sense. There were progenitor cells in the lymph node. They seem to get activated after treatment. They seem to give rise to these intermediate exhausted cells. And I've told you that the expansion of those intermediate exhausted cells in the blood has been previously shown to associate with clinical response to treatment. So we then wanted to know, is the lymph node response that we see correlated with an expansion of those cells in peripheral blood? And we were fortunate to be able to collect blood samples from these people as well before treatment and at the surgical time point, which is after treatment. And so we again analyzed the CD8 T cells that we had seen in circulation from these individuals. And we found that there were two populations or clusters of CD8 T cells that significantly expanded between the baseline and surgical time points. Those two clusters were both consistent with an intermediate exhausted phenotype. So they no longer expressed TCF1, still expressed PD1. One of these had higher expression of Ki67, that's cluster 5, and the other cluster 9 had higher expression of granzyme B, which would be consistent with more of an effector-like state. So we think probably these Ki67 positive cluster 5 cells are becoming the cluster 9 upregulate granzyme B. We also saw in the blood that those intermediate exhausted cells, the cluster 5 cells, had increased their proliferative rate. More of these cells were Ki67 positive in the blood after treatment. And that correlated really well with what we saw in the lymph node, but it was, of course, possible that cells were getting activated in the tumor and then leaving the tumor and entering the blood. And so we wanted to ask, do we see evidence that these same subsets are proliferating in the tumor? And the answer was pretty conclusively no. We could find these Ki67 positive cells in the blood, but when we looked in the tumor, they really weren't proliferating there. We did see them, they just weren't expressing Ki67. So again, that would be consistent with this idea that the response in the lymph node allows cells to become activated, expand, enter the bloodstream, and ultimately go to the tumor to carry out their effector function. Now, in this analysis, I've shown you in a couple slides that we also analyzed metastatic lymph nodes, and I haven't shown you any of the data yet. And so we wanted to know, does the presence of metastasis in the lymph node actually impact any of this biology? So we started by looking at the abundance of different T cell subsets in either uninvolved lymph nodes that don't have tumor metastases, or in paired metastatic lymph nodes from the same individuals where there was cancer cell invasion into the lymph node. And we found that there were a variety of subsets of T cells that were differentially abundant. Again, the clusters that were more abundant in the metastatic lymph nodes were terminally exhausted clusters, and the clusters that were more abundant in the uninvolved lymph nodes were the progenitor exhausted clusters. And this might make sense because now antigen is actually present within the lymph node itself. And so you might imagine those cells would be more likely to differentiate due to chronic antigen stimulation. So what happens to these progenitor cells after treatment in metastatic lymph nodes? Do they still respond properly? And we found that they don't respond in the same way. So if we looked at these TPECs that were still there in the metastatic lymph nodes, we saw that they were less likely to generate clusters of intermediate cells around them after treatment. That's shown here on the left. So you can see in the uninvolved lymph nodes, there are more intermediate exhausted cells surrounding each TPECs, and we don't see that in the metastatic lymph nodes. So that tells us that they fail to expand in the same way. And we also saw that they don't upregulate PD1 in the same way. So the TPECs and the intermediate cells in the uninvolved lymph nodes had upregulated the expression of PD1 consistent with TCR signaling, but again, that did not happen in the metastatic lymph nodes. So we then wanted to know, where are these cells in the metastatic lymph nodes? Are there cancer cells that are directly surrounding them, maybe preventing them from getting access to antigen-presenting cells? That hypothesis wasn't true. The TPECs that were still there were still in regions of lymph node that looked like normal lymphoid tissue. And so we analyzed their neighbors in a bit more detail to see what else might be different. And we found that the dendritic cells that surround TPECs in metastatic lymph nodes have upregulated a variety of different immunosuppressive molecules, including CD39, IDO, PD-L1, and TIM3. So the antigen presenting cells are in a more suppressive state surrounding these TPECs in the metastatic nodes. And there were also Tregs surrounding these TPECs in the metastatic nodes, and they had also upregulated expression of molecules consistent with better suppressive capacity, including FOXP3, the master regulator transcription factor, as well as TIM3, that's a marker of Treg suppressive capacity. So that suggested that these TPECs, while they might still be there in small numbers, become surrounded by immunosuppressive cells, and that could be a reason why they fail to expand after treatment. So if that were true, you might expect that patients with metastatic lymph nodes would also have a smaller circulating T cell response. And that again is what we saw. So if we looked at patients that either did or did not have metastatic lymph nodes, the patients with lymph node metastasis had less expansion of those intermediate exhausted cells in the blood, and they were also less proliferative after treatment. And this data is very consistent with clinical data in lung cancer showing that patients with lymph node metastasis are less likely to respond to checkpoint inhibitor treatment. So just to summarize this part of the talk, hopefully what I've shown you is that in regional lymph nodes, there are progenitor exhausted CD8 T cells that are enriched, and they're clonally related to the more terminally differentiated exhausted cells that are present in the tumor microenvironment. Immune checkpoint blockade activates these TPECs, but it leads to a reduction in their frequency and their differentiation into these more intermediate exhausted states. This in part happens because TPECs co-localize with dendritic cells, and those cells then upregulate activating signals. And in metastatic lymph nodes, both TPECs are reduced in frequency and their responsiveness to immunotherapy seems to be diminished, perhaps because they become surrounded by these immunosuppressive niches. So with that understanding, I want to tell you a bit about some unpublished data from the lab as well, thinking about what might go wrong after immunotherapy. Here I've shown you that the response initiates in the lymph nodes. What might cause tumors to be resistant to immunotherapy beyond some of the mechanisms that we already know, which are largely genetic, loss of either antigen presentation or the interferon gamma signaling pathway? So this was work done by a postdoc in the lab, Namu Cho, who's just actually started his own group at UCSF. And what Namu came to the lab interested in understanding was this question of why are some tumors, even if they might have a lot of mutations, still resistant to immunotherapy? So we know that there's an association between tumor mutation burden and response, meaning that tumors that have more mutations presumably have more potential for neoantigens that can be recognized by immune cells and therefore are more likely to be responsive to checkpoint inhibitors. But even for highly mutated cancers like microsatellite unstable colorectal cancer, which have a ton of mutations, about half of the patients are still resistant to treatment. So why might that be the case? Presumably there are plenty of antigens that the system can see, but there's still not a productive response. So this was the question that Namu set out to tackle. And he did this by making a variety of different syngeneic mouse models that were either microsatellite stable or unstable by knocking out MSH2, and then asking whether the microsatellite unstable tumors responded better to checkpoint inhibitors. So this is our data from the B16 melanoma model, which has been used very widely to study tumor immunology. And this looks pretty much like what you would expect. The microsatellite unstable tumors grow out a little bit more slowly. That's comparing the light blue to the black lines. And after they're treated with immunotherapy, we do see some response in the microsatellite stable line, that's shown in the dark blue curve, but we see a more profound response in the microsatellite unstable line shown in the pink curve. So indeed, having more mutations and microsatellite instability sensitizes this model to checkpoint inhibitors. But the other half of the models that Namu made looked something like this, where regardless of whether they were treated, untreated, microsatellite stable or unstable, the tumors grew out at exactly the same rate. And so here, we really wanted to know what's the underlying biology that's driving this type of resistance. And these tumor lines have a similar number of predicted neoantigens from whole exome sequencing. Those antigens are present at a similar variant allele frequency. So it's not as though they're only present in like 2% of the cancer cells. And both of these models, the responsive and the resistant ones, express MHC class 1 and upregulate it further in response to interferon gamma. So it's not either loss of antigen presentation or loss of the interferon gamma signaling pathway. So something else is going on here. So Namu performed CyTOF analysis on these tumors to see, in terms of the tumor microenvironment, what were some of the major differences that he observed between the responsive and the resistant models. And the biggest thing that stood out was that there was a massive change in the neutrophil infiltration within the responsive and resistant models. So in the resistant model, we saw that there were more neutrophils present at steady state. That's the purple group. And that after the tumors were treated with checkpoint inhibitors, that frequency actually increased even further. That's the light green. Neutrophil production in the bone marrow is governed by the growth factor GCSF. We saw that induced after treatment in the resistant tumors. And their chemotaxis into the tumor microenvironment is regulated by CXCL1. And we also saw more CXCL1 being produced after treatment. So in these resistant models, we were seeing more neutrophils accumulate, and that was associated with elevated levels of these soluble factors that govern neutrophil expansion and chemotaxis. And we found that the major source of these molecules were tumor cells and fibroblasts in the tumor microenvironment. Tumor cells outnumber fibroblasts about 10-to-1 in this model. So we think the dominant source is probably the cancer cells, but the fibroblasts certainly contribute as well. Now, neutrophils have been previously shown to be immunosuppressive in a wide variety of cancer contexts, particularly immature neutrophils, sometimes called myeloid-derived suppressor cells. And so we asked that question by isolating these neutrophils and putting them in co-cultures with T cells, and we found that indeed they do suppress T cell proliferation. So these are a suppressive subset of neutrophils that we see expanding. Now, Mu then wanted to know, what might be driving this biology, and we had previously found in the lab that in this tumor model that's resistant, there's a high production of GCSF, and that's actually dependent on an upstream cytokine called IL-1, which needs no introduction to the Denver immunology community with Charles Gianarello here. And so we'd found that by blocking IL-1, that GCSF and CXCL1 production were actually abrogated in this tumor model. So that's the upstream driver of these factors. And so Namu asked, if we block IL-1 receptor, could we actually sensitize these models to checkpoint inhibitors? And in fact, that was the case. So if we gave checkpoint inhibitors with an IL-1 receptor blocking antibody, the tumors grow more slowly. We've sensitized them now to the checkpoint inhibitors. We found there was also similar biology in another tumor model here. This is a MOC1 model of head and neck cancer, where we also see between responders and non-responders that the major difference is the production of these soluble factors and neutrophils. So the working model so far is that IL-1 is produced here. We see that it's mainly produced by tumor cells, also by fibroblasts, and that IL-1 can act in an autocrine fashion through NF-kappa B. I haven't shown you that data, but if we give an NF-kappa B inhibitor, we block this response as well. And that's not surprising. It's the major signaling pathway downstream of the IL-1 receptor. And GCSF and CXCL1 and 2 are NF-kappa B target genes. So this is the model. Tumor cells produce IL-1. They can signal to it in an autocrine fashion through NF-kappa B. That drives the production of CXCL1 and GCSF. And those recruit neutrophils to the tumor microenvironment, which shut down the T cell response. That all seemed plausible and like it could make sense, but there was something kind of missing from our understanding here. And that's that these checkpoint inhibitors mainly act on T cells. And so presumably, because we see this biology amplified after treatment, there must be something the T cells are doing to actually drive this resistance mechanism. Either that, or the checkpoint inhibitors are acting on another cell type that we're unaware of. And so we asked, what might the T cells be doing to actually contribute to this inflammation-driven resistance program? So we started by looking at the phenotype of the T cells between these two models, the responsive one and the resistant one. We found that there are T cells in the resistant tumor, first of all, actually at a pretty similar frequency to what we see in the responsive model. So here's showing all of the different groups that we compared. The important comparison is the pink group, which is the MSI high B16 responsive model treated with checkpoint inhibitors. And if you compare that to the green group, this is now the resistant model treated with checkpoint inhibitors, you can see that they have a very similar number of CD8 T cells in the tumor microenvironment. So it's not a failure to recruit T cells to the tumor. But the T cells that are in the tumor are very different between these two contexts. So here we performed, again, a CyTOF analysis and clustered the CD8 T cells and asked what states we find these cells in. And in the responsive model, we see a lot of effector-like cells that are proliferating and making granzyme B. I don't think my cursor, oh, it is working here. So you can see those accumulating down here at the bottom of this differentiation trajectory. In the resistant model, we see that these cells kind of get stuck. They don't differentiate into those effector cells. So they're still present in the tumor, but they haven't upregulated granzymes and they're not proliferating to the same extent. So the T cells look different, but they're there. Are they the driver of this resistance mechanism? And so to answer this question, Namwu depleted the T cells using either an anti-Thy1 depleting antibody or anti-CD4 and CD8 depleting antibodies, and asked, do we still see neutrophil expansion, GCSF production, CXCL1 and 2 production after the immunotherapy is given? And the answer was no. If we deplete the T cells, we prevent the amplification of that resistance mechanism. So the T cells seem to be dependent or required to amplify the inflammation-mediated resistance. So we thought this was pretty interesting because this is now the first mechanism we're aware of where effector T cells could actually drive resistance. It's a bit paradoxical. So we wanted to know then, what are the T cells doing to drive resistance? And I've told you that a lot of the underlying biology here is dependent on NF-kappa B signaling in tumor cells. And so we thought, well, the obvious candidate would be that T cells might produce TNF-alpha, which also signals through the NF-kappa B pathway. And there were old studies in tumor cell lines showing that if you stimulated them in vitro with IL-1 and TNF-alpha, that they actually act in synergy. So we thought maybe that's a possibility. And that's consistent with what we saw. So if we looked in the tumor microenvironment after treatment, we saw in the resistant tumors, there are more TNF-alpha producing T cells. And if we did co-cultures between those TILs and tumor cells, we could see that GCSF really isn't produced at very high levels if you only have one cell type or the other. If you have both cells together, now you get a lot of GCSF production. So the CD8 T cells can elicit GCSF production from the tumor cells. But if you now add an anti-TNF blocking antibody into the culture medium, you can see that that reduces the GCSF production from the tumor cells pretty considerably. And when we looked at the effect of stimulating our tumor cells with either TNF or IL-1, we could see that the combination did indeed result in synergistic production of GCSF and CXCL1. So the next idea would be maybe if we get rid of the TNF production from T cells, we could make the response better, if that's the underlying mechanism. And so to test this, Namwu first did experiments in TNF knockout mice where the whole host lacks TNF, but the tumor cells, of course, still retain the capacity to produce TNF. And he saw that TNF knockout mice, now the same resistant tumor model became responsive. So in the absence of TNF, we could restore responsiveness in this system. Now, TNF was lacking in a lot of different cell types, and so we wanted to know, is it actually dependent on T cell TNF as opposed to other sources, such as fibroblasts or myeloid cells? And so Namwu crossed CD4 Cree mice to TNF flox mice, so now only the T cells have no production of TNF. Now, because T cells go through a double positive stage in the thymus, this actually deletes TNF from both CD8s and CD4s, even though it's a CD4 Cree, so it's all T cells here are lacking TNF. And in the system, again, we see that now the resistant model becomes responsive. So if the T cells cannot produce TNF, they can't drive this inflammatory resistance mechanism, but they still can carry out their effector function. And in response, the tumors are better controlled. So what about in patients? Is there any evidence that similar biology might be taking place? Namwu is a physician scientist. He's a radiation oncologist. And so he had the inspiration to go back to the medical records from patients treated at UCSF who had microsatellite unstable tumors. So all of these people have highly mutated tumors. and could look in the radiology reports to see whether patients were classified as responders or non-responders after treatment. And because complete blood counts are done as part of routine clinical workflows, we actually could see the number of neutrophils and the number of lymphocytes in the blood from these patients before and after they were treated. So the idea was, if it was consistent with the mouse model, in the resistant patients after treatment, you might see an expansion of neutrophils in the blood. And that is pretty much exactly what Namu saw. In the patients that progressed on treatment, there was an increase in the neutrophil counts in peripheral blood, but in the non-progressing patients, there was no statistically significant change in neutrophil counts. So that tracks pretty well with what we're seeing in the mice. And then Namu asked, if we go back to clinical data from patients treated with immunotherapy, could we stratify patients based on their expression of this inflammation signature together with CD8 T cell infiltration? So we know that you need CD8 T cell infiltration to respond to immunotherapy. So the T cell cold tumors are expected to be resistant. And that's what we see in the blue and the green groups here. These patients have pretty low overall survival. This is patients with lung cancer treated from the Genentech OAK study with anti-PD-L1. So that leaves the CD8 high patients, the patients that actually do have T cells. And if our hypothesis were correct, then this inflammation resistance gene signature that involves IL-1 receptor expressed by the tumor cells together with NF-kappa B, CXCL1, GCSF, if we combine those into a gene signature, if you had a high expression of that gene signature, we would predict that you might be resistant to immunotherapy. That group is shown here in the yellow curve, and you can see that their overall survival looks pretty much the same as the people who don't have T cells at all. And what we would predict would be the subset of patients who do derive benefit from treatment are those who have T cells, but don't have this inflammation signature. And that's what we see. The subset of patients that have improved overall survival are those in the red curve, again, that have T cell infiltration, but that don't engage this inflammation-mediated resistance circuit. And so to summarize this part of the talk, hopefully what I've shown you is what we think is a mechanism by which effector T cells can actually drive resistance to immunotherapy through their production of TNF-alpha. That TNF-alpha can act on tumor cells and probably also on fibroblasts to synergize with IL-1 in the tumor microenvironment. This also requires IL-1 receptor to be expressed by the tumor cells. So if we knock out IL-1 receptor on the tumor cells, the biology goes away. And so they need to be able to respond to both TNF and IL-1. And in doing so, they drive a lot of NF-kappa B signaling, production of NF-kappa B target genes like GCSF and CXCL1. And those now recruit immunosuppressive neutrophils into the tumor microenvironment, which shut off the T cell response. So this is a negative feedback circuit, essentially, induced by the T cell to shut off the T cell. And if we intervene at various stages along this mechanism, we can actually improve responses to checkpoint inhibitors. So with that, I'm going to wrap up, acknowledge everybody in the lab who led these studies. The first project on the lymph node responses was led by 3 amazing postdocs in the lab, Trinalina Olcom, who's a computational biologist, Kyle Jones, who was a pathologist, now works at Genentech, and Maha Rahim, who's an experimental biologist and got the MIBI platform set up at UCSF. She works at a small company called Enable Medicine now that is making spatial profiling technologies available to folks, as well as all of our clinical collaborators with whom there's no way we would have been able to do any of the work that we had done. I'm not a physician myself, and so it's I consider it a real privilege to be able to collaborate, with folks who are actually treating patients and working together to define clinical trial study designs that allow us to learn as much of the biology as we can in a way that's, of course, hopefully also improving patient care. The study participants and their families, without whom, of course, there would have been no clinical trial, and who are really willing to partner with us on these types of initiatives. And then the second project I told you about was led by Namu Cho, who's now an assistant professor at UCSF and is actively recruiting for his new lab. So for the students out there, if that biology was exciting to you, reach out to Namwu. And then the funding sources who pay for all the work. And with that, I'm going to wrap up. I was also asked to put this slide up here for a CME survey. So if you feel like telling me how my seminar was, go to the QR code there and let me know. And thanks so much for your attention, and happy to take any questions.
Questions? Beth?
Are we taking questions from online yet?
Oh, yes, thanks. So the question was about the lymph node biology and whether the suppression of T cell responses in the metastatic lymph nodes may be due to higher antigen load. And if that were true, then you might expect that there would still be responses in more distant lymph nodes where tumor cells have not yet metastasized. And the answer is we are doing those studies now. It's hard to do in the patients, obviously. The lymph nodes that we looked at were paired metastatic and non-metastatic from the same individuals, but they were generally from the same neck levels that are expected to have the same sort of drainage. So I think it's consistent with that idea, like that model is still a very real possibility that there's more antigen in the metastatic nodes, so that leads to more terminal exhaustion of those cells. But it's not yet one that we've proven as such. So we're doing studies now in mouse models where there is reproducible metastasis to the lymph node to try to get at that question where we can actually toggle some of these variables. But I do favor that hypothesis as being a relevant one. So I'll keep you updated.
And then my second question is, have you looked at kind of the particular networks in the metastatic themselves?
Both really interesting. So the first question is about Tregs. Are the Tregs required for suppression of the TPEX, and do we see them moving to distant sites? And the answer to that, I'm going to pull from data from one of our collaborators, Nate Redeker-Flynn at Stanford, who's looked at this question in some detail. And what he's seen is that antigen-specific Tregs are induced in mouse models of lymph node metastasis. And he sees those Tregs migrate then to the lung, where they can help see distant lung metastases by suppressing T cell responses in the lung. So that does seem to be the case. Whether they're required formally for TPEX suppression, we don't know yet. But again, we're going to do those studies by depleting Tregs in the context of these lymph node metastatic models. And then the second question was about the reticular network in the lymph node and whether that might be altered. And we haven't looked at that yet in great detail. We know that there's not a large difference in the number of fibroblastic reticular cells that are surrounding the TPEX in metastatic or non-metastatic lymph nodes. We did look at that, so the number of neighbors isn't changed. We thought that would be important because the TPEX actually express pretty high levels of IL-7 receptor, and the fibroblastic reticular cells are the main source of IL-7. So we thought there might be good reason for them to be co-localized. We do see them in proximity, but it's not different between the metastatic and non-metastatic lymph nodes. But we need to look at that, generally in more detail and understand, perhaps we're getting more of like a CAF-like signature, cancer-associated fibroblasts in the metastatic lymph nodes compared to the uninvolved. So don't have an answer for you yet, but definitely something we're interested in too.
Mercedes?
I'm curious on the first one. I'm curious about the detection of the T cells proliferating in peripheral blood in the blood. How do you envision that the cells proliferate while they are just in circulation? Or what triggering the release of the cells according to your theory that the cells come from the lesion. None of the cells that are in the lesion, they don't get released. So I'm just puzzling by the fact that you see high frequency, almost like a tumor cell that continues proliferating even in that.
Yeah. So the question is about proliferating enables upregulation of S1PR1 and allows the cells to leave the lymph node, but they probably just haven't degraded all of the Ki67 protein, so we're still able to stain those cells as being Ki67 positive. That would be my suspicion. I don't think they're probably still actively undergoing cell division in the blood, but that's something we should look at, and there would certainly be ways that we could.
There was also a question on Zoom. What happens if you add exogenous GCSF?
We have not done that experiment, but it's very clinically relevant because oftentimes after chemotherapy, patients will have neutropenia, and GCSF is actually given to rescue neutrophil counts in cancer patients. So we haven't added it. It would be important to do. We have taken it away, and if you take it away, then the tumors become more responsive. It looks very similar to, the blockade of IL-1 or the other factors, TNF, the other factors that I showed you in the system. So we think that adding GCSF is probably going to have a detrimental effect, but we haven't shown that conclusively. And of course, it is a very translationally relevant question.
And the last question on Zoom, are the immunosuppressive dendritic cells in the lymph node resident DCs, or have they migrated, or immunosuppressive DCs that have migrated to the metastatic lymph node?
The short answer is we don't know yet. We don't have great markers to distinguish those two in our MIBI data sets, and so that's something that we'll be getting at in more detail in the mouse models as well.
I'd like to thank Matt for being here, and thank you for being here. Yes.